Hello guys, so today we are going to discuss about the histology of muscle. So first we have muscle tissue. It is a fourth basic type of tissue with epithelium, connective tissue and nervous tissue. Okay, so we have three types of skeletal muscle. We have three types of muscle tissue, that is skeletal muscle, cardiac muscle and smooth muscle. Okay, so skeletal muscle is composed of very long multinucleated cells with cross striation. This is important as cross striation is important. Okay, their contraction is quick, forceful and usually under voluntary control. Okay, voluntary control is important as the examiner might ask that skeletal muscle is involuntary or involuntary control, and if you say involuntary control then you are going to be F. Okay, so cardiac muscle, it has a cross striation, it also has cross striation. Okay, but it is involuntary. Okay, and the special property of cardiac muscle is that they have their cells, they are interconnected via the intercalated discs. Okay, okay, contraction is involuntary and vigorous and rhythmic. Okay, smooth muscle, they are collection of fusiform cells, like the cells, they are spindle shaped. Okay, and they lack striations and they have slow involuntary contraction. Okay, so in all types of muscle, contraction is caused by sliding interactions of thick myosin filaments along thin actin filaments. Okay, and the plasm of this muscle tissue as sarcoplasm, and endoplasmic reticulum of smooth muscle as the sarcoplasmic reticulum. Okay, and the membrane of the plasma membrane of the smooth muscle as the sarcolemma. Okay, fine. Then talking about skeletal muscle, the formation of skeletal muscle is from the mesoderm. Okay, mesoderm, they form the myoblast and they eventually differentiate into myotubes, and these myotubes then proliferate and give rise to the muscle tissue. Okay, and the some cells, they don't differentiate and they remain adjacent to the myotube satellite cells. Okay, and these satellite cells are important as whenever there is a muscle injury, then to get new tissue, new muscle tissue, we need this. Okay, so medical application is that hypertrophy and hyperplasia. Okay, hypertrophy is when we do exercise, like when we do exercise, then the myofilament, actin and myosin filaments in the muscle tissue, they grow, and that's called the hypertrophy. Okay, it is basically the increase in volume, in cell volume. Okay, cell volume increase. Okay, that's all hypertrophy. Tissue growth by increasing number of cells, tissue growth by increasing number of cells is the hyperplasia. Okay, this is hyperplasia. Okay, okay, fine. Yes, this is, as you can see, they are myoblast. They form myoblast fusion to form myotubes, and myotubes, they differentiate and form the muscle fiber, and this is satellite cell. So organization of skeletal muscle. So see, there are three connective tissue layering around the skeletal muscle. So first one is the epimysium. It surrounds the whole muscle and it is composed of thin, dense, irregular connective tissue. Okay, why it is called dense? Because it has more collagen fibers than the number of cells. Okay, like the collagen fibers are synthesized by the fibroblast. Okay, so the fibroblast number is less as compared to the collagen fibers. Okay, and okay, this is the epimysium. It is an external sheath of dense irregular connective tissue surrounding the entire muscle. The entire muscle term is important in this. Okay, okay, and nice. That's okay. Now perimysium, it is a thin connective tissue layer that immediately surround each bundle of muscle fiber, termed a fascicle. Okay, fascicle is important. Fascicle is important. Perimysium, it surrounds the fascicle. Okay, and what is fascicle? It is a bundle of muscle fibers. Okay, and yeah. Nerves and blood vessels and lymphatic penetrate the perimysium and supply each fascicle. Okay, fine. And endomysium it is an external lamina of individual muscle fibers. In addition to nerve fibers, capillaries form a rich network in the endomysium, bringing oxygen to the muscle fibers. Okay, you can read this and you can understand. It's simple. Okay, epimysium, perimysium, and endomysium. Okay. Epimysium surround the muscle, perimysium surround the fascicle, and endomysium surround the endomysium surround the external lamina of individual. This epimysium, perimysium, endomysium, and with the dense irregular connective tissue of deep fascia which overlies the epimysium. It is continuous with the tough connective tissue of tendon at myotendinous junction is basically a junction where the muscle is attached to the tendon. Okay, muscle is attached to tendon, bone or skeleton. or skin. Okay, so so sarcoplasm is highly organized and consists of long cylindrical filament bundles called light bands are called eye bands, and they consist of solely, they contain only actin filaments. Okay, actin filaments. And yes, and the actin filaments, that they are attached to the z disc, and the distance between two z discs is called sarcomere. Okay, and yes, and the M line is the line which is present in the A band. Okay, A band, where the actin and where the actin filaments are not present. Okay, as we can see that the A band contains both the actin and myosin filaments. Okay, but the portion of the A band that does not have I band is called the H zone. Okay, and the middle of the A band is called the M line. Okay, and here in the M line we find the also contains the myosin binding protein called myomycin that holds the thick filament in place. Okay, myomycin is a protein that is present in the that holds the myosin binding that holds the thick filaments in place. Like do not get displaced during contraction. Okay, okay, so yeah. And the creatine kinase, it is an enzyme that catalyzes the transfer of phosphate groups from phosphocreatine, storage form of ADP. Okay, phosphocreatine, it has a wait yes, it is composed of two identical heavy chains and two pairs of light chains. Two pairs, listen carefully, it has two pairs of light chains. Okay, two pairs means four light chains. Okay, I was very confused regarding this, and I don't want you to be confused. So yeah, two pairs means four light chains. Okay, and two heavy chains. Okay, two identical heavy chains. Okay, myosin heavy chains are thin rod like motor protein twisted together as myosin tails. Okay, now see, these myosin heavy chains, they form tail of the myosin. Okay, heavy chain, two identical heavy chains form the tail of the myosin molecule. Okay, fine. And the globular projection of containing the four, see, here they have mentioned four and here two pairs. Okay, so don't get confused. Okay, four myosin light chains form a head at one end of each heavy chain. Okay, so what is the takeaway? See, there are four chains, two heavy and four light. Okay, two heavy chains form the they form the they form the tail of the myosin molecule, and four light chains, they form the head of the myosin. Okay, fine. That's it. Okay, and yes, filaments are it is also called the actin molecule, and actin molecule is composed of three parts, that is F actin, tropomyosin, and troponin. Okay, so F actin is a it is made up of G actin, which is a monomeric actin. Okay, and tropomyosin is a long two polypeptide chains. Okay, it is composed of two polypeptide chains. Okay, and troponin is a complex. See, as you can see here, this is the tropomyosin, this is troponin, and this is the actin monomers. These actin monomers are called G actins, and when they combine, they form the F actin. Okay, fine. And troponin, a complex of three subunit T and T. Okay, troponin it is composed of three parts: troponin T, troponin C, troponin I. Okay, troponin T is responsible for the uh, for the for for the binding of tropomyosin to actin. Okay, F actin. Okay, and troponin C it is responsible for the binding of calcium, and troponin I regulates the actin myosin interaction. Like it is like a bodyguard, like security. Okay, like a TNI. Okay, fine. I band consists of positive fan, that is not drawn. Okay, which is okay. So see, actin filaments are anchored perpendicularly to on the Z disc by. Okay, so see, actin filaments are joined to the Z disc via the alpha actinin. Okay, and and the remind me, myosin molecules are bind to the Z disc via the titin molecule. Titin is a protein. Okay, it is responsible for the binding of the this myosin molecule to Z disc. Okay, myosin to Z disc, titin, and actin to Z disc is called as a alpha actinin. Okay, and okay, and here it is given titin. Okay, and another large accessory protein, nebulin, bind each thin myofilament. Okay, so see, myofilaments are actin and myosin. Myofibril is basically the whole length of the cylindrical portion of which consists of sarcomeres. Okay, reticulum and transverse tubule system. So sarcoplasmic reticulum is the endoplasmic reticulum of the smooth muscle fibers, smooth muscle cells, and yes, and see, transverse tubule, it is basically invagination. See, long finger-like invagination of cell membrane sarcolemma, it forms transverse tubules. Okay, and these transverse tubules, they form triad, like the finger-like projections of this sarcolemma, they extend into the sarcoplasm, and they lie adjacent to the terminal cisternae of the smooth end of the sarcoplasmic reticulum. Okay, and they form the triad. See here, two terminal cisternae is called triad. Triad commonly allow depolarization of sarcolemma into tubule to affect the sarcoplasmic reticulum and trigger calcium release of calcium ions into cytoplasm around the evening part and at the in the evening. Okay, fine. Bye for now.